carbon is utilized in formation of 2 molecules of ribose 5-phosphate and 4 molecules of xylulose 5-phosphate. This ribose 5-phosphate is used for formation. This is the sugar. This is the pentose sugar which is required for nucleotide synthesis of nucleotide, synthesis of nucleoside and nucleic acid. This is the sugar base of nucleic acids. Okay, nucleic acid. Nucleic acid has sugar base, then sugar, then a uh, uh, then adenine, guanine, or cytosine, or thymine. Of this any one will be required for formation of the nucleosides. Then from nucleotides can be produced by the addition of phosphate. Okay, so we have not mentioned what is the use of this NADPH. This NADPH, it has various. It is used for reductive synthesis of some important biomolecules, as for example, fatty acid steroids like this. We will be mentioning full list when we cite examples. Okay. So from this ribose 5-phosphate and xylulose 5-phosphate again condensed together to produce fructose, 5 molecules of fructose 5-phosphate. This fructose 6-phosphate, 5 molecules of fructose 6-phosphate, it can enter into the Emmons-Meyerhoff's pathway of glycolysis. Is it clear? Is it clear? We are showing this is the complete, more elaborate figure. Glucose 6-phosphate, 6-phosphogluconate, then ribulose 5-phosphate, 1 molecule of carbon dioxide is reduced. If there is 6 phosphogluconate, there will be 6 carbon dioxide. Then ultimately ribose 5-phosphate, xylulose 5-phosphate produces. We have seen in the previous figure. Then it is acted on by transcatolase, producing this 5 and 5 molecule carbon and 10 molecule of carbon used to produce pseudoheptulase, which have 7 carbon and glyceraldehyde 3-phosphate. Okay, 3 carbon. Then again they condense together under the influence of enzyme catolase, transcatolase, producing fructose, this 7, 3, 10. Okay, they condense together to produce fructose 6-phosphate, erythrose 4-phosphate. Fructose 6-phosphate, we know 6 carbon, erythrose 4-phosphate has 4 carbon. This is the diagrammatic representation. Okay, next one important figure that I mentioned to show you. You see this glucose 6-phosphate from which is the substrate for hexose monophosphate strand pathway. So if this is the point of starting of glucose oxidation, this glucose 6-phosphate by this alternative pathway produces fructose 6-phosphate that enters into the Emmons-Meyerhoff's glycolytic pathway, or if it produces glyceraldehyde 3-phosphate, it produces this. It can produce this glyceraldehyde 3-phosphate. Okay, These are the possible. So the hexose monophosphate shunt pathway, the products that is produced at the end, fructose 6-phosphate that enters in again into the Embden-Meyerhoff's glycolytic pathway, and it can also enter into the glycolytic pathway as glyceraldehyde phosphate, 3-phosphate. Okay, this is for interest. Now come to the point. So this figure, I think this is a bit elaboration of this short outline. This one is good for citation in the exam, also for Viva. Okay. Comparison of Embden-Meyerhoff's glycolytic pathway with pentose phosphate pathway. If we compare these two pathways, it will be more easier for us to remember. Substrate for Embden-Meyerhoff's glycolytic pathway is glucose. Substrate for glucose 6-phosphate pathway or pentose pentose 6-phosphate pathway. This glucose 6-phosphate. Glucose 6-phosphate is the substrate for pentose phosphate pathway. Okay. In glycolytic Embden-Meyerhoff's of glycolytic pathway, ATP consumed. Also, there is production of ATP. But in pentose phosphate pathway or hexose monophosphate shunt pathway, ATP is not produced. No ATP is produced. The coenzyme of Embden-Meyerhoff's glycolytic pathway is NAD, nicotinamide adenine dinucleotide. The coenzyme of hexose monophosphate shunt pathway is NADP, nicotinamide adenine diphosphate. There is one phosphate, addition of one phosphate, but that differs the coenzyme. In glycolytic pathway, it is NAD, and hexose monophosphate shunt pathway, the coenzyme is NADP. Occur almost all tissues. Glycolytic pathway occur in almost all tissues. It occurs in liver, red cells, adipose tissue, adrenals, adrenal cortex, gonads. In glycolytic pathway, carbon dioxide is not produced. Carbon dioxide is produced in hexose monophosphate shunt pathway. Carbon dioxide is again produced in the TCA cycle. In TCA cycle, if we recollect, in TCA dicarboxylic acid cycle, citric acid cycle, carbon dioxide is produced, but in Embden-Meyerhoff's glycolytic pathway, carbon dioxide is not produced. Is it clear? Carbon dioxide is produced in hexose monophosphate shunt pathway. The similarity between these two pathways, Embden-Meyerhoff's glycolytic pathway and pentose phosphate pathway, is that both the reaction takes place in cytosol. Both the biochemical reaction takes place in cytosol. This is an important similarity among a lot of dissimilarities. Okay. We'll be then proceeding to importance of pentose phosphate pathway a bit. Let us remember synthesis. Pentose phosphate pathway is very much important for synthesis of ribose, pentose sugar. This ribose is utilized for formation of nucleoside, nucleotide, and nucleic acids. Nucleic acids, that means DNA, RNA. Coenzymes, reduced coenzyme is produced, that NADP is produced in pentose phosphate pathway are used for synthesis, reductive synthesis of fatty acid, sterols, steroids, bile acids, nitric oxide. Very important biomolecules. These are okay. It is NADPH, reduced NADP is required for synthesis of fatty acid, sterols, steroids, bile acids, nitric oxides. Maintain the reduced state of glutathione for antioxidant activity. Very important. Again, it is required for phagocytosis by the WBC. It is also NADPH is also required for drug metabolism. These are the important uses of pentose phosphate pathway. Pentose. The main byproducts is ribose and the NADPH. They are used for production of these important biomolecules for which body is very dependent on this pathway. Okay, okay, okay. Hello. এখন না পরে okay. If glucose is stored as glucose within the cell, then it will disturb the osmolality of the cell. That means within cell there will be hyperosmolality. That's why glucose is stored as glycogen. Is it clear? So glycogen is the polymer of glucose stored mainly in the liver and muscles. Most of the two-third of the glycogen is stored in muscles and 1/3 is stored in liver. Is it clear? This liver glycogen is very important in the maintenance of the blood glucose level. Muscle glycogen cannot participate in the maintenance of the blood glucose level, but it can serve as sources of energy. Is it clear? Synthesis and breakdown of glycogen in the liver during the synthesis of glycogen occur in the liver during feeding state and breakdown of glycogen occur during fasting periods. They play a very vital role in the maintenance of the blood glucose level. Liver is called glucostat because immediately after meal, when blood glucose level becomes very high, it lowers the blood glucose level to normal level, and when in fasting period, in between meals, when blood glucose level is decreased below the normal, then it elevates the blood glucose level. So that's why liver can be termed as glucostat. Okay, it play very important role in the maintenance of the normal blood glucose level. Okay, we mentioned this amount is arbitrary. If your height is more, body is bulky, storage of glycogen will be more, and if it is thin, it could be less. This figure is from our figure Orientals. In Western figures, the storage of glycogen could be more because they are more bulky. Usually, they are more bulky. Regulation of blood glucose level is very important. Liver glycogen, because of the presence of why liver glycogen is more important than muscle glycogen. Liver glycogen, because of the presence of enzyme glucose 6 phosphatase. This glucose 6 phosphatase we can make it bold liver glycogen because of the presence of glucose 6 phosphatase can convert glucose 6 phosphate to glucose, and muscle glycogen because of the absence of glucose 6 phosphatase. Glucose 6 phosphatase muscle glycogen is absent in glucose 6 phosphatase. That's why glycogen can't be converted to glucose. So muscle glycogen can't participate in maintenance of the blood glucose level. This is very, very important point to note. Is it clear? Why? Now in exam, this is our paid question. Why liver why muscle glycogen cannot participate in maintenance of the blood glucose level, or what is the advantage of liver glycogen over muscle glycogen? These are the doable explanations. Right? Is it clear? Next, our time is passing, so let us be a bit fast. Synthesis of glycogen from glucose is called glycogenesis. Glycogenesis is the synthesis of glycogen from glucose. Is it clear? Glycogenesis is the synthesis of glycogen from glucose. That means for glycogenesis, glucose is the substrate. It occurs during abundance of glucose in the blood. Abundance of the glucose will be possible when immediately after meal. Immediately after meal, there is blood glucose level increases in our blood. Okay. Primer is essential for glycogen synthesis. Without primer, is not glycogen synthesis is not possible. Primer, I think, this is the source. This is the teeny fragment, small fragment of glycogen over which glycogen can be synthesized. Just for preparation of curd, if we wish to prepare curd, there must be some source of curd a bit. Okay, otherwise it is not possible for synthesis of the curds. Pre-existing glycogen fragment can serve as primer. When it is absent, it is synthesized by glycogenin. It should be synthesized by glycogenin. Is it clear? Synthesis of glycogen is enhanced by insulin. It is enhanced by insulin. So it occurs during immediately after meal. Immediately after this person is entering just before the end. What is the use? Glycogen synthesis, and this is outline of glycogen synthesis. You see glucose is converted to glucose 6 phosphate. The same reaction that happens in glycolysis enzyme is mostly glucokinase, GK, glucokinase. Also hexokinase. ATP is converted to ADP. That means there is expenditure of energy. For phosphorylation of glucose, glucose 6 phosphate is converted to glucose 1 phosphate. Enzyme responsible is phosphoglucomutase. Phosphoglucomutase. It serves. This step is both way. This enzyme can convert glucose 1 phosphate to glucose 6 phosphate. Also convert glucose 6 phosphate to glucose 1 phosphate. Here in glycogen phosphate, the glycogen synthesis, glucose 6 phosphate is converted to glucose 1 phosphate. This is just isomerization of glucose 6 phosphate to glucose 1 phosphate. That means phosphate group in glucose 6 phosphate was present on carbon 6. in glucose 1-phosphate, phosphate group, it is transferred to carbon 1. Uridine diphosphate is required for glycogen synthesis. This uridine triphosphate conjugate with glucose 6-phosphate, forming uridine diphosphate glucose. Is it clear? For glycogen synthesis, there are two requirements. There is one glucose, another is uridine triphosphate. The third is ATP. Okay, this uridine diphosphate provides glucose to the glycogen fragment, to the primer, and glycogen chain is elongated. Okay. Okay. The enzyme responsible is glycogen synthase. In this pre-existing glycogen fragment, the uridine diphosphate glucose transfers glucose one by one, yeah, and gradually elongation of chain takes place. Is it clear? And branching enzymes is also help in this glycogen formation. Is it clear? And with the help of these two enzymes, glycogen synthase and branching enzyme, which is under the influence of insulin, there is synthesis of glycogen. Glycogen synthesis occurs when there is abundance of glucose in the blood immediately after meal. Okay. Next. So our next point is synthesis glycogenolysis. Glycogenolysis is the breakdown of glycogen to glucose. It occurs during fasting and in between meals. In between meals. Is it clear? Remind one thing: this glycogenolysis, this is not a reversal of the glycogenesis, but this is another reaction which is responsible for conversion of glycogen to glucose. Glycogen to glucose. Is it clear? Glucagon and epinephrine enhance glycogenolysis, and this glucagon and epinephrine is more in fasting periods. Or when body requires more energy. The process occurs mainly in the liver and muscles. This is the outline of glycogenolysis. You see glycogen. The main enzyme for glycogen synthesis is phosphorylase. Is it clear? Another enzyme, helping enzyme, is debranching enzyme. Then this two enzyme is responsible for conversion of glycogen to glucose 1-phosphate. This glucose 1-phosphate then converted to glucose 6-phosphate. Enzyme responsible is phosphoglucoamidase. This enzyme is same for synthesis of glycogen. That means the phosphoglucoamidase is a common enzyme for synthesis of for glycogenesis and glycogenolysis. When glucose 6-phosphate is produced, glucose 6-phosphate in the liver, this is important to remember. Glucose 6-phosphate converted to glucose. Glucose 6-phosphate is converted to glucose. It occurs in liver. In muscle, glucose 6-phosphatase is absent. That's why muscle glycogen cannot be converted to glucose. And in muscle, glucose 6-phosphate that is formed is utilized for energy purposes. Is it clear? Gluconeogenesis we cannot start because there is less than 1 minute. What to What to note from this gluconeogenesis? You can see on this outline. That is also good. In next class, we will be discussing on this gluconeogenesis